These RNA viruses lack DNA entirely and replicate their genetic material using RNA-dependent RNA polymerases. Furthermore, the discovery of epigenetic modifications added another layer of complexity to the central dogma. Epigenetics refers to heritable changes in gene expression that occur without alterations to the underlying DNA sequence. The cellular dogma incorporates epigenetic modifications, non-coding RNAs, protein modifications, and cellular metabolites as integral players in the intricate web of cellular information. It recognizes that the cell is not merely a passive recipient of genetic instructions, but an active interpreter and responder to its environment. The cellular dogma expands the language of life beyond the traditional trio of DNA, RNA, and proteins. It recognizes that information within a cell is stored, transmitted, and interpreted through a diverse array of molecules. Non-coding RNAs, once dismissed as junk DNA, play a crucial role in regulating gene expression. These RNA molecules, including microRNAs and long non-coding RNAs, can silence or enhance the translation of specific messenger RNAs. Protein modifications such as phosphorylation, acetylation, and methylation act as molecular switches that can alter protein activity, localization, and interactions. These modifications add another layer of complexity to the proteome, the entire set of proteins within a cell. Cellular metabolites, the small molecules that are the intermediates and products of metabolism, are no longer viewed as mere bystanders in the cellular symphony. They act as signaling molecules influencing gene expression, enzyme activity, and cellular responses to environmental cues. Section 5. A multidimensional flow information dynamics in the cellular dogma. Unlike the unidirectional flow proposed by the central dogma, the cellular dogma embraces the complexity of information flow within a cell. It recognizes that information flows in multiple directions, forming intricate feedback loops and regulatory networks. Gene expression is not solely determined by the DNA sequence, but is influenced by a multitude of factors, including epigenetic modifications, non-coding RNAs, and cellular metabolites. These factors can act as switches, amplifiers, or dampeners, fine-tuning gene expression in response to internal and external cues. The cellular dogma acknowledges the dynamic interplay between different cellular compartments. The nucleus, once considered the sole repository of genetic information, is now seen as one node in a complex network of communication that includes the cytoplasm, organelles, and the extracellular environment.